Friends, in the previous module, we have seen the general aspect of quality control. The next criteria in quality assurance is functional aspects, mainly resistance to gastric condition, bile acids, adherence and colonization of intestinal epithelium, antimicrobial activity against potentially pathogenic bacteria, and modulation of immune system. So, first we will see resistance to gastric condition. Probiotic bacteria must be able to survive in the gastrointestinal tract. The survival of ingested probiotics in the different parts of gastrointestinal tract varies with the strain. Some strains are rapidly killed in the stomach, while others such as strains of bifidobacteria or lactobacillus acidophilus can pass through the whole gut at a very high concentration. Lactobacilli are more acid tolerant in the food carrier. On the other hand, food matrix determines their pH tolerance. The pH of gastric environment is 1.2 to 2.5. The bifidobacteria, however, proved less acid tolerant than the lactobacilli, particularly when exposed to human gastric juice. Second aspect is resistance to bile acids. Probiotic organism must be resistant to bile acids. Bile acids are synthesized in liver from cholesterol and secreted from the gallbladder into the duodenum in the conjugated form. These acids undergo extensive chemical modifications in the colon. In relation to the assessment of the probiotic strain intended for the human consumption, the most relevant determination is that ability to grow in the human bile. Next functional aspect is adherence and colonization of intestinal epithelium or tissues. Probiotic adhere and colonizes. This adhesion of probiotic to intestinal mucus and epithelial cell has long been considering one of the most important selection criteria for probiotic microorganisms. Adhesion to the intestinal mucosa may prevent the probiotic cells being washed out and therefore enabling temporary colonization, immune modulation and competitive exclusion of pathogens. In order to produce enzymes, lactic acids, vitamins and natural antibiotics, the probiotic strain must be adhered to intestinal wall, colonize and multiply. Probiotic strain isolated from the human gut adheres and colonizes better than probiotic strain isolated from animal origin. The next functional aspect is antimicrobial activity against potentially pathogenic bacteria. The probiotic strain should be capable of producing antimicrobial substances is most important in developing probiotic supplement and probiotic rich foods. Several metabolic compounds produced by lactic acid bacteria have antimicrobial activity. However, bacteriocins or proteinaceous substances with specific inhibitory activity against closely related species are most studied. At present, Niacin is the only purified bacteriocin approved for use in the product intended for human consumption. Niacin is active against stain mainly Staphylococcus, Micrococcus and Listeria species. Apart from this, other probiotic strains are also produce variety of bacteriocins which have adverse effect on various pathogenic microorganisms. The next functional aspect is modulation of immune system. Strains of probiotics should be able to stimulate as well as regulate a several aspects of natural and acquired immune response. There is a significant difference between the ability of bifidobacterium and a lactobacillus strain to influence the functioning of the immune system appears. Probiotics are suggested to confer the protection against enteropathogen by stimulating the cytokine production, enhancing the phagocytic capacity of polymorphonuclear cells and macrophages, enhancing the specific antibody response to the pathogens. Hence, so far we have seen functional aspects of quality assurance where we have discussed resistance to gastric condition, bile acids, adherence and colonization of intestinal epithelium or tissues, antimicrobial activity against potentially pathogenic bacteria, and lastly modulation of immune system.